Anita.
Oh, good. Okay, we should probably get started.
Thank you, everybody. Here it is.
Sorry about that. Whoa.
Well, I'll turn off mine. There we go. Approve the audio.
Yeah.
So just tell everybody to type their questions if they have them.
Yeah, I'll go ahead and do that. So we're really sorry if there's no. Oh, I see what happened. Let me make you a host. That's what happened because you don't have microphone rights. Okay. So again, I think Marianne, you want to try again? Okay.
All right. Oh, yeah. Okay, that should do it. There were two microphones I had to. So anyway, I'd like to introduce Anita Bandrowski, who's the project leader for the Neuroscience Information Framework. My name is Marianne Martone. I'm the PI. And today we're going to have an introduction to NIF's latest release, NIF 4.5, including the new beta interface that we have made available for your feedback and testing, which has a lot of new features available for searching across the NIF. Anita will give you an overall introduction to what the NIF project is for those of you who are new to this. But basically, the Neuroscience Information Framework project was designed to make it easier for researchers, educators, administrators to search across and find relevant resources, largely produced by NIH funding, but not exclusively, because we recognize that a lot of content and things that is being produced is not easily searchable through engines like Google or through the literature via PubMed. So NIF has set up various indexes and to make it easier, search functions to make it easier for researchers to find resources. And by that we mean data, materials, tools, services, basically anything that a researcher would be looking to find. So I'm going to turn this over to Anita. I'm going to mute my microphone since we are in the same room. And also, generally, if microphones come on, we get feedback. So we ask you to stay mute until the end. However, if you do have a question, please type it into the chat window and I will let Anita know and she will address it. At the end, we'll take general questions. So let me turn it over to Anita.
Okay, thank you, everybody. Whoa, can you mute your microphone? Thank you.
That was kind of cool.
That was very cool. So is everybody hearing me okay? Okay, well, oh, okay, great, wonderful. Thank you very much. That's great. All right, so let me minimize this screen, and you should be able to also see my screen now. And I think that I will just. Oh, okay. Okay. Oh, I think this is pretty good. All right, so First of all, welcome and thank you so much for attending. I think this is a great new tool. We'd like to unveil it with you today, and what I want to do is just go over some of the new features, but please feel free to type in any questions that you have. I'll go ahead and stop because this is going to be a live demo. So what I want to do is just show you, this is the main website of NIF, and what I'm going to try to do first is type in my favorite gene. And not many people know about this feature, but if you type in gene : and then your favorite gene name here, which is in my case GRM1, not sure why that's my favorite, but that's always my favorite. But in any case, I just wanted to make sure and point out that everyone understands that when we get these kind of a structured query where I am telling the NIF system that what I'm looking for is a gene and the name that I know is GRM1, that the NIF system actually goes ahead down here in the View Edit Query box and expands to all of the known synonyms of GRM1, which is the metabolite glutamate receptor, 1A, and all of those. And just very quickly, I mean, this is something that you've probably been familiar with. Okay, so there are new people. So if you what you can do is you can go ahead and move these things back and forth. But one of the things that you'll end up with this query is you'll end up with a lot of the categories of the data sources that you have here on the left. And so, for example, the category is called nervous system level genes. And what we've done is we've taken a lot of different databases. We're up to about 170 altogether. And it looks like there are about 20,000 results for GRM1. So essentially, some of those come from the Allen Institute. This is the Mouse Brain Atlas, and you can look at the gene symbol here. These are all the same gene, but they're expressed at different densities and levels in various parts of the brain. So this is one way you can look at this. Here, a set of results from Amigo. This is a large consortium of many of the model organism databases, which actually tell us information about this gene and its functions. So these are, for example, you can know more things about some of these genes that you might be asking about. There are obviously interactions from BioGrid. And then for NIDA researchers, this particular database might be particularly interesting because this is a database largely made up of supplementary material from various literature sources that all deal with some kind of a dependency publications. So there's a lot of nucleus accumbens literature in here, and most of these, although not all of the results, are from microarray studies. So we know that those supplementary tables have tremendous amounts of data, and we've been able to thankfully liberate some of those and structure them in some reasonable ways. In any case, I'm not going to go through every one of the data sources, but you can kind of flip through and see, you know, what these different things are, these different databases. They do have a little bit of a description at the top for all of them, and then they have a way to get back to the original source. This is a source that you should be probably familiar with. This is homologene coming from NCBI, and this just gets you back to the National Library of Medicine telling you more information about the homology of this particular gene. So if you click out onto any of these, usually it's in the first column, that takes you back to the source itself. Okay, so there are many of these different nervous system levels, and we also have data types like antibodies against GRM1, plasmids with GRM1 genes associated. These come from Addgene. But these are all the kinds of things that you can do. One of the things that you may not be familiar with is that you can actually search within a particular column. So you can do sort. So clicking on this little arrow will allow you to sort and it will actually allow you to find and search within that column. So this is a way that we've made available some of the capability from our back end. So here I can actually search for just human, and in this case it won't change the results very much, but this is one way that we've actually allowed searching to be a little bit better. You can also export most of the results. You can open them in a new window just to get a better view of the full thing, because sometimes you know, there are many, many hundreds or thousands of results. So this is sometimes a very helpful thing. Okay, now let's move over to the literature. And what I wanted to do is just show you a couple of the new features inside of the literature. So here we have this, you'll be familiar with being able to look at the essentially the title and whatnot. If you click on the little blue link, it does take you to PubMed. I won't show you that. But this particular gene, GRM1, or any of its synonyms, are found in these particular sentences from this paper. And in this case, the paper is actually has been annotated by OMIM. So if you click on the little link out button, you can get to a particular database, a particular project that actually looked at that paper and said something reasonable about that particular paper. So again, here we can we link out to, again, this is OMIM, but this will be different depending on whichever particular link out you might be looking at. So this is one of the things that we've enabled. And I think Gordon just asked a question, but I missed what it was.
How do I get Sam?
Oh, can you handle that on the chat box? Okay. Okay, so one of the things that I wanted to draw your attention to is now there's this little section box. And the section box actually gives you the ability to take this particular search term and search, let's say, only within the methods section of these papers. And when you do that, you can go in and look at the various papers that are actually mentioning your particular search term inside of just section, right? So this is kind of a new way of being able to search for this. And here we can see that we can go not only to PubMed, but also PubMed Central. Now, we don't have the full text for every one of the papers, so this PubMed Central and this annotate button will only come up where we do have access to the full text. And what I wanted to show you is, in addition to these link outs, what you can do is you can actually help us annotate some papers. So here I'm going to open up the annotation tool. And this is the Domio annotation tool coming from Harvard University, Tim's lab. And Tim is actually sitting right next to me, so it's good that this is working. So, you know, this is a wonderful tool, and it's a wonderful tool because it allows people to say things about, here, let me make this a little tiny bit smaller so I can actually see. Okay, there we go. So what it allows you to do is that it actually allows you to say things about this paper. So you can take portions of this paper and say, no, I agree with this, I don't agree with this. But the things that we're interested in are allowed when you actually are able to look at some of the types of annotations. So Domio already goes ahead and extracts out the bibliographic information. So as you're going through Domio, or through and looking at a paper, you actually see that these are the list of, this is the list of publications that's associated with this particular paper. And what we care about quite a bit are the methods. So let me actually just show you how to do something that's kind of cool. Here's a single paragraph out of the methods section, and what I'd like to do is actually like to text mine that. So I'm going to, I hit the text mining button and I hit the analyze button. And now what it's doing is it's asking the NCBO web service to go ahead and recognize certain terms about this particular paper that from the section that I have actually highlighted here. So I can look at things like embryonic, but that's probably not. So tissue culture. Well, here's a mouse monoclonal antibody. So here's a monoclonal antibody, and the nice thing about the tool is it gives you the list of different matches. And what you can do is you can say, yes, I agree with this one. This is actually the correct. This is the correct thing. If you actually click on the, if you go ahead and click on this term, then it will give you a bit more information about this. And I'm sorry about the code of this. We should probably maybe link that to something a little more human readable. But in any case, so it does give you some information about this particular item. The other thing it can do, and let me just kind of kill this for now so we can see what we're looking at. So the other thing that we can do with this tool is actually look at a particular antibody. So here we've got, this seems like a reasonable antibody. So we've got this PYK2 CAK beta mouse monoclonal antibody from BD Transduction Laboratories in Canada. Okay, so what we can do is we can actually go ahead and annotate that. And annotate. Okay, good. And this is an antibody. We can go ahead and check off which subjects, which species. But one of the, oh, let me pull my screen up a little bit. All right. So here, I can actually say this is the PYK2, and let's just try to search for this antibody. Now, we don't have a catalog number, but maybe, just maybe, we can actually find this antibody, and let's see what happens. So the idea here, and the reason that I'm only doing BD, because actually BD Transduction Laboratories will not come back because a lot of these vendors do change their name and people don't necessarily get this right. So the authors may call it BD Labs, BD Transduction Labs, and many, many variants. But here we actually get back different antibodies from BD Biosciences, which is actually the correct vendor, by a different name. And now we're not completely sure because we don't have the catalog number here. And these are all different catalog numbers. So we can't be sure which antibodies this is. So I'm actually going to go ahead and not add this because I don't know which of these antibodies this paper is using. In another case, I actually looked this up. If we look at the phospho-PK2, all right, so there's a better situation here with another one of these antibodies. Again, the problem is if we don't have this, okay, so here's phospho-PK2 from cell signaling. And so if we say cell signaling technology is the vendor, we have no catalog number, we have a name, and it's this name here. And let's just see.
We have a question from Sri Joy as to whether the antibody registry relatively complete, because I've typed it in.
Right, so this is coming from the antibody registry. the antibody registry is relatively complete. It's not perfect. It has about 200 individual vendors in it, and there is some duplication because the vendor will change, and sometimes we'll have just multiple antibodies. But in any case, the registry is about as good as you're probably going to be able to get. It's as good as any of the commercial vendors. So here is the antibody that we're looking for. It is from Cell Signaling Technology. And again, if you have the catalog number in the paper, this makes this process much easier. But I'm going to go ahead and apply this. Okay, and so now I can actually see that I have this antibody. And so now when I go through the rest of this paper and maybe want to annotate some of the nice figures, I'll be able to reuse this antibody and tag it into any particular part of the paper that I want to use. So and so what I want to be able to do eventually, if this, when this makes it through the curation pipeline, that this is in fact a valid annotation on top of this paper, then what we'd want to be able to do is we'd actually want to be able to search some of these annotations. So let me just show you one of the new ways that we can actually search annotations. So here's a little annotation database that we've been able to put together. And what we can do is we can either search by the publication number. So let me redo this search. And back. Here we go. And so you can either search by publication number, or you can search by the type of entity. So here I want to look for antibodies, for example. Here I'm still searching for mGlu-R1, which is the search that I did before. But what you can see is that I know that this particular antibody from Sigma Aldrich was actually used in this publication. And in most cases, those can also be found under the link out portion of the of PubMed. So we actually allow other people, including PubMed, to grab these annotations. We expose them ourselves. But wouldn't it be cool if we could get a large body of these annotations and then be able to ask the question, where was this reagent used? How many other people used it? And then under what circumstances? This isn't fully available right now, but the more of these annotations that we're able to gather from the literature, the more then we can expose and give back to PubMed as, yes, the author said this is exactly what this is, this is annotating. Okay. So a couple of other searches that I just wanted to show you. So I'm going to put the results, make the results a little small, and just show you a couple of things. If I search for any particular term, I can just search for hippocampus. First of all, the NIF allows you to autocomplete, tries to autocomplete. Here it tells you that the hippocampus is an anatomical structure. You can find out more information about it if you click the Neuralex button. The Neuralex is just going to tell you what we think the hippocampus is, right? So there's a definition, and then it actually does many things. Does a search of the databases for every single term, and will tell you lots of things, including, where does this brain region hold? This is at the mammal. It is a regional part of the brain. It is part of the hippocampal formation, et cetera, et cetera. And it has these very interesting synonyms that are not necessarily lexical variants. And let me show you what we do with those. So I'm going to search now for the hippocampus, and you can see here that the hippocampus and all of its synonyms come out and actually go ahead and search the data and the literature and, of course, the registry. But what we can also do is we can actually look at this parts relationship. And here I'm going to select all of the subparts, and that goes ahead and redoes the search with all of the subparts also of the hippocampus. So you can do this for most of the structures. The other thing that I wanted to show you is another type of query. So every once in a while you're going to get these terms that are bold. And here I'm just starting to type in drug, I'm letting it autocomplete. And drug of abuse is actually sort of a monster query. So the National Institutes of Drug Abuse have labeled certain compounds as being abused substances. So what NIF has been able to do is basically to take all of these substances, take all of their trade names and all the other various types of synonyms, and we've added these to a large list, which then we search across the literature, the data federation, and everything else. Now, you should probably appreciate that this is a very long list. This is a monster query, so it usually takes a little while for the data to actually come back. But in this case, even with these 1,000 or so terms, because you can appreciate all of these drugs actually have quite a few synonyms and trade names. This actually gives you a pretty good list of all of the papers. Oh, I'm still searching methods, I believe. Yeah. Well, you can do that as well. So you can know that there are at least 178,000 papers where any drug of abuse is mentioned in the methods. So again, this is a relatively good search tool, which and here are the grants. Okay, I'll bring this down. All right, so with that, I think I want to, oh, the Data Federation did come back, and there's quite a bit of data for any of the drugs of abuse. I'm not sure why this is blue. I think I clicked on something funky. Okay. Yeah, but here, right, exactly. So here's where you can actually see a lot of the drugs of abuse. So see if I can put up there are various things that we can, oh, it's just, not sure why that's blue. All right, so going back, and the other piece that I wanted to show everyone is this beta interface. So if you actually just click on that beta portion, that little window, you'll see our new interface. And what we're doing here is we're doing a lot of things with these filters to narrow down the amount of information that we actually bring to you. So you'll end up in this data tab. And the data tab will actually show you and present the individual databases in a bit of a different way. So what you're seeing here are the names. So here's ABCD, and they have, there's a lot of information about brain regions because this is the avian brain surgery database. And then the plasmid database are alphabetically ordered, so this arguably is at least a different way of looking at a lot of this data, and possibly a better way. So here we have brain info, brain-machine interface, etc. Jonathan is raising his hand. Jonathan, is there a comment that you'd like to make? Oh, he's saying sorry. Okay. All right. So one of the things that I'd like to show you here is on the left, there are all these search filters. So you can actually go ahead and look at just the connectivity, or you can look at the activation foci, or you can look at any of the other data sets, biospecimen, for example, and then only those particular things will come back. And then to clear the filter, you just push the little X here, and that gets you back to the full list. So what we can still do is still search for any of our favorite terms, like, for example, oh, one thing that I wanted to show you, the terms and conditions. So one of the things that we've also done is we've brought in a lot of the information about the database, including its terms and conditions, and we're working on these pages to try and make this a little bit more informative in terms of when was it last crawled, when did we get it, how fresh is the data and all of that. But at least this gets you to the terms of conditions and also where you would find those on the individual database website where we have that information. You can get it. You can also see all of the results. In this case, this is for AdGene, so you can do the same thing here just to browse the data. You can also search the data. So let me actually show you what that looks like. Nope, just kill that and come back. All right, so now let's search for cerebellum because we like the cerebellum. It is our favorite brain region. And let's just look at the Allen Mouse Brain Atlas here. And so one of the things that we've always had a problem with in NIF is that there aren't the specific ways to say, I'm looking for the brain region cerebellum. Because see, when you...
We have a question whether the new interface will coexist with existing interface.
So the new interface for at least some amount of time will actually coexist, hopefully happily, with the old interface. And if you click any search and on this classic NIF, it will take you back to the normal interface. This we're rolling out as a beta product right now to kind of get feedback, and that's why the annoying feedback box is always appearing. If you get feedback, we promise that it should go away for about a day. But if you do not give us feedback, then we'll keep popping up the annoying feedback box for probably the next couple of months, just because we want to ascertain whether people really like this better or whether they don't, because we'd like to make a decision, and we'd like that decision to be informed by the users. So what I wanted to show you is that we've always had this problem where if a gene name is called, zinc finger protein of the cerebellum, then that piece of data comes back. But the beautiful thing now is that we can actually look at this, and one of the ways that we can do this is we can look at these facets on the left-hand side. So here under structure name, we actually see that there are some results for cerebellum, there are some results for cerebral cortex, hippocampal formation, etc. And so now we can just click on the cerebellum here, and now the only structures are cerebellum. And it does so happen that the zinc finger protein of the cerebellum still is the top result, but that's all right. You can click on the gene symbol and actually get this thing to sort alphabetically or reverse alphabetically. But the structure name you can see here is always cerebellum. So this tells you what you're actually doing. So you can actually get rid of that, or you can get rid of the ascending search and get back to your same results. Another way you can do this is we're actually going to type in anatomy. So there are these, I'll show you those categories in a second, but one of the other things we can do is the same thing basically by saying, okay, I know that the cerebellum that I'm asking for is not the gene. I would like to complete the search only in the anatomy column, which in this case is called structure name, but in other data sources, the column might be called anatomical region, it might be called some other things. In any case, the thing that I'm looking for is the anatomical region called cerebellum, and so now again we have the same results, the structure name, only cerebellum, and let me get rid of that annoying box. Okay, so this is the thing. There are other things you can play with here, like you can export the results as a CSV, so you can always get this on your laptop. You can look at them in different ways. Sometimes this doesn't. It's sort of depending on how you want to look at this. But let's go back now, and let me show some of those others. So I've just clicked the data to kind of go back to where we were before. And you can see here that these are these queries that you want to try out, or you could try out. You can type in cell, you can type in disease, or you can just click on these things. And here's my favorite gene, so molecule GRM1, and you could see again that here are the results. So there are 101 results from Amigo, there are some results from the antibody registry, etc. And again, these things can be aligned by data type, etc. Okay, so roughly, I would like to now kind of get a feeling for, what are the other things that people are have questions about and would like to ask about, because there are many things that I can show you. I can show you some of the services. What was that? Oh, yeah, I should absolutely show my NIF. So we can actually save searches. So one of the nice things is that you can actually register or log in. I do happen to have a login. So there's my login. And when I log in, okay, I didn't try this one. I don't, well, I should be able to. I think it's just, okay. So, no. Oh, because I'm in Firefox. In Chrome, I'm already logged in. Okay, so what I can do is I can reset my password. I can see my saved searches. So there are things that I can actually do here. I can store papers. I can save searches. So apparently this login has no saved searches. All right. I do have some stored papers, things that I'm interested in, and you can continue to, you can explore this if you like. You can search by the new papers, where later on what we'll do is we'll possibly send you a NIF digest if you choose to have this. And the NIF digest is actually a set of literature based on these papers that you've chosen that are presumably interesting to you, including, so there are papers. We tend to pick out grants that we think might be interesting, some social buzz, etc. We're actually getting ready to release a new version of this in the next couple of days. So if you are already a subscriber, then we will send that to you. All right. So now again, I think we've had some questions from the,
Can you set it, so we have a question from Erin.
Okay.
The question.
So the question is, can you set it to run a saved query periodically and mail you updates? Okay, that's, Mary, thank you for that question. I'm going to go ahead and promote everybody to presenter. That way you can go ahead and ask and turn your microphone on if you like. However, what we don't have that at the moment. So it is definitely one of the things that we have planned to do, but we cannot do that at this point. So you're actually going to have to go in and rerun that query on your own. Does that answer your question, Mary? Yeah, okay. So I'm making everybody present. I think there's a way to do this en masse. But
However, you may want to
go ahead and I've killed my microphone. Or my...
Hello, can you hear me?
Hello, can you hear me?
Jonathan said. So with the NIF digest,
Jonathan
if
said.
you sign
So
up for
with
it,
the NIF
it asks you to set up your interest there, and automatically
Set up
every
your
month send
there.
you papers that meet your interest profile.
Paper.
And one of the things we want to be able to do is to search our data sources and other things as
One
well.
of the things we want to be able to charge as well. Those are our plans.
Okay, so everybody should be able to have micro rights now. Give us a query. Jonathan seems to be typing because apparently he doesn't have any. He has no microphone. Ah, cert zebrafish. All right, let's try that. I'm wondering if cert is a gene. Nope. Cert zebrafish does not bring back anything. It probably brings back some literature. But it does not bring back data. Yeah, let's try it. Should be able to know. No, there is something. 2 results, not a whole lot. Just tells you a little bit about this gene and not much more. So when you deselect zebrafish, then you actually get a lot more data, including antibodies. But apparently these are not specific to zebrafish specifically.
There's very little known about.
Yep.
Only 17 papers.
Well, if you open it up to CERT by itself, it's almost 2,000 papers.
Mm-hmm.
But that's still a pretty small amount of data. Okay. What if you changed to homology name?
What about the structure of the cell?
What do you mean, Joy, about
Maybe molecule.
structure? Ah, okay, so we can try.
That would do fewer.
Yeah, that should do fewer, not more. But the SL, what is it? SLC6 is probably where we want to go. So let's try gene colon SLC6. Oops, 6. Okay, let's try that. No, it doesn't bring back. So it actually, there's very little known about this gene because homologene doesn't. Well, no. There's more for SLC6, including some gene expression from various organisms and UniPro. Okay, so that actually helps, Jonathan. And you've got a better, it's the 4B, is that what you're looking for? Okay, so John is asking, where do we find the search categories? Okay, so the search categories, one of the things that you should be able to do is in the old NIF, you should be able to use gene colon, and the place where you can actually find most of these is right here in the new beta interface. We haven't made all of these things available through the classic NIF interface, but through these, you should actually be able to say molecule colon. It would help if I spelled molecule correctly. Okay, let's see. And that was SLC6. Yeah. All right, that brings back all the results. And actually, that's about right. That is about right. So it gives you all the results from, for example, Amigo. Here, there's 860.
And together.
You can certainly. Sorry, you probably didn't hear that. So the question was, can you and these things together with species? So largely yes. Unfortunately, from Amigo, we currently don't have the species listed here. So we only have the taxon identifiers, which you can see they are. This is Homo sapiens, but this is a data issue where we just haven't quite gotten to setting these taxon IDs for the species.
We have a question from John.
Okay, John. But can we look up the categories that are available for narrowing the search? Yes. So there are multiple ways to do that. The way to do that here, I think the most reasonable way to do that is to go ahead and look at the new interface, and you can actually look at the different kinds of things that you can do in terms of the facets. So the facets will actually tell you, for example, which ontology namespaces appear in the results section. So for example, we know that there are 301 results for molecular function, 296 for biological process, and then these are the different ontology terms that are actually being brought back here. So, an evidence code, for example. So what I wanted to then do is, for example, look at serotonin uptake, and these are all of the statements. Go away, annoying box. These are all of the statements for that are by the different entities here about, I believe this is human. Yep. About the particular gene. So, does that answer your question? Jonathan, i.e. gene question, species
Yeah,
question.
I just tried.
Yep.
But maybe ask John to turn on his microphone to make sure we're getting.
Yeah, John, can you turn your microphone on your computer because I'm not sure if I'm getting your question. Okay, he seems to be typing.
If he doesn't have a microphone.
He might not have a microphone. Okay, what are the search qualifiers? What do you mean, Jonathan, by search qualifiers? So my search...
Oh, it doesn't turn on. Okay.
Okay. So your search qualifier is, so the search qualifiers are molecule here, and you can also do organism, you can also do anatomy, and the way that you see those is you click on the main data, and you can actually see them here. So you can look at protocols, phenotypes, organisms, and so far this is basically what we have. Yep.
Yeah.
It, yeah.
Hi. So this is
Marianne. Okay, the search categories that we have available reflect the modules in the NIF ontology. We just had a noting from Andrea that gene isn't listed. Genes are actually a special case because they don't come from the ontology so much. We've always gotten all our gene names from just the NCBI gene service, and so that part needs to be re-implemented. Right now, we kind of lump all molecules together, including genes, under the molecule category, because we find when we go through these sources that researchers will often use gene as a substitute for a protein or a of a part of a protein or an RNA probe. That is, they're not very specific. So we're experimenting with substituting molecule for gene. One thing that the new beta interface does not do yet, however, and it's a little bit confusing, and we apologize, is that gene query that Anita showed you in classic NIF does not currently work in the NIF beta interface. That will come back. Should come back in the next week or so, we hope. And we've been debating whether gene stays as a special category or whether, again, if the way that most
All right, are there any other questions? Are there any other genes or Shreejoy is typing? So let's see what we get. And again, please feel free to. Okay, so Srijoy is asking, when you do a literature search, is it just sending a query to PubMed? I think that the answer is no. In fact, there are a couple of things that we do that are pretty different from PubMed. So we do search the PubMed corpus. What was that? But Jeff is telling me that there's a local index, which is the case. But we do much more than just search for the PubMed results itself. Because what you can do is you can actually, search within PubMed Central results, the open access portion of PubMed Central, in addition to searching the PubMed results. So you can also see different things, like you can search within paper sections here, just like you can in the classic NIF interface. And you can actually sort your results, and you can know how many results there are per year. So this is kind of we think, a useful feature, just to kind of give you an idea of, when this particular gene was studied or this term was being used. The author facets are maybe not quite as good in terms of figuring out who's saying it, but sometimes there's a big bolus of papers from one particular group, so this should be able to tell you. And the other thing that we do is, we ask our annotations, whether anybody has said anything, and in most cases there are no annotations on top of these papers, but where they do exist, we will bring them back. So those are the three different indices that we actually hit for the literature. Okay. And Marianne was saying that we also pass the full NIF query string to PubMed so that we can search for synonyms. Yeah.
Typically, we're searching the literature.
Right. So we are, and you don't see this much in the new interface, but actually where there are in the classic interface, you can actually see this much more. Although this particular example is bad. So if we look at our hippocampus, GABAergic neuron is a good one. Right, so here we're looking for GABAergic neurons. And again, this is one of these monster queries. And GABAergic neuron is actually translated, if you look, into all of the neurons that we know of that actually have been labeled as using GABA as a neurotransmitter, and all of their synonyms. So again, this will take a little bit of time to actually come back as a query, but you can see that the hippocampal basket cells, globus pallidus principal neurons, dentate gyrus cells, etc., are all here, and you can see the full extent of these GABAergic neurons. And again, if there's something that we've missed and you want to help us actually go ahead and bring this up, you can always open this up into Neuralex, and Neuralex is a wiki. That's not good. It's the one we don't have information on. Let's see if there's a better one. Okay, and you can usually get back to that particular cell, and you can actually hit the edit button and give, for example, a definition of this cell, an extra synonym of this cell, and you can give us other properties, shape and whatnot. And again, these are all tagged with the person who actually made the note, and these get curated and fed back into our ontology, which then helps us inform this next query. Okay, so Srijoy is saying thank you, and Manu is typing.
Go through some of those results,
Okay, yeah. So again, you should be able to know in bold here what we've actually found, and here we're just looking at neurons, GABAergic neurons, Purkinje cells, Lugaro cells, etc. So as we kind of scroll down, we're looking and we're finding these specific neurons that are in fact GABAergic cells and not, example, thalamic reticular neurons. So this is one way where you can ask a kind of a different kind of query, right? Because what you're really saying is, do you have this type of thing as opposed to do you have this specific thing?
So Manu is asking about an API or script.
Yes.
For fetching information.
Right, so the API and the scripting language and all of that, if you actually look into the main site, oh, there's an outage notice. That is tonight, okay. So one of the things that you will see is you can find a section that's called For Developers, Manu. So let me read the question because I'm not sure if everybody heard. So do you have an API for or a scripting language to fetch the information directly from the program? And the answer is yes, absolutely. For the ontology information, that's the information that you know, a GABAergic neuron translates into these things. You have these ontology services, and there are, I think, 9 different ones that we've, no, 12 different ones that we have exposed publicly. So get concepts, search parents, search siblings, etc. And then for database query, so every single one of these, the new interface is completely built on top of these services. So Manu, you should be able to get what you need, and I will address specific questions if you like. Give me, drop me an email, and I will be happy to go ahead and help you with your query, because there are a lot of different services that that are here, including searching, for example, a summary service, which is right here. And so you can actually see that the Q equals gives you the query. All right, you're welcome. Okay, John is typing. Okay. Okay, so while he's typing, it's very helpful. Oh, right. Okay, so about the Neurolex, this is one thing that was brought up from this room. So the Neurolex has been led by Dr. Gordon Shepherd. There's an effort there to actually name these neurons very consistently, and to also do some interesting things in terms of the properties of these various neurons. So, for example, when we see the olfactory bulb accessory or main cell, there's actually a set of properties that was agreed upon within the PONS subgroup of the International Neuroinformatics Coordinating Facility, which actually had agreed upon the naming system, the structure, the types of properties that are filled in. And so every single one of these forms is actually something that was agreed upon and is being led as an international effort, but primarily by Shepherd. And the types of properties that are here, for example, the density, the branching types, the types of somas, sizes, locations, etc. These are all structured fields that are actually being pulled, as a function of the intellectual output of this group, which is very valuable because this makes these things machine readable. So helping us, the Neurolex representation actually makes this more available for text mining. So that should hopefully help in terms of this. Okay, are there any other questions or yeah. Okay. So, as I was showing you before about the terms and conditions, there are tutorials and there are search videos and other things that we have tried to make available for, a lot of these sources. There's a page on each of these sources, and each one is actually linked back to the source itself. So here's a non-for-profit source that redistributes plasmids, and there are typically tutorials for each of these, letting people know sort of what are the kinds of things they can search for and where they're going to find these results. Of course, this is all in the classic NIF, and we haven't made too many tutorials. A few of the videos for the new NIF interface. So primarily, this is the way that we get back to this. So generally, hopefully, this will help you get a better understanding of what are some of the things that we actually have. We hope that the lists of databases, the way the way that we actually, what are the sources that we have, how many results do they have, and what do these results look like just to browse. There are now these pages of these summary results in the new beta interface that should be available. And yes, we will post a link to the recording of this webinar very shortly, and hopefully we'll also put it up on YouTube just to get a broader distribution. And I would like to, at this point, thank everyone very much. Apparently we didn't have many people who were able to turn on their microphones, but thank you so much for typing in the questions diligently. And we hope to help. So we are really trying to help the researchers in neuroscience primarily, but generally. And if we don't do something, or you can't figure something out based on the tutorials that we have, we are always here. You can hit the feedback button, and we will actually get back to you with some emails. And Jonathan is telling us that he is also on Twitter and Facebook and all of these other Google+. So please become our friend if you like. And again, we do monitor those sites. And Pinterest. I don't know.
I don't think we're on there yet.
I don't think we're on Pinterest. I think I thought that was only a recipe thing, but
No,
for whatever you want to group. Okay. Yeah, Jonathan should get on that because he is our guru and substantially younger than us. So Pinterest. Okay. Yeah. Okay. Well, in any case. Again, thank you very much for listening in, and please hit the feedback button. It's here, so you can let us know and submit any feedback. And if you leave your email address, then we'll be happy to get back to you. Okay. Thank you very much, and have a good day. Have a good rest of your day. Okay.